All right, so last time we went through the cell cycle, the parts of the cell cycle, and mitosis, how cells make exact copies of themselves. G1S, G2, mitosis, cytokinesis, and then the whole thing starts all over again. However, we know that cells are not reproducing all the time. Some cells are reproducing faster than others, some aren't reproducing at all. So today's focus is going to be on how the cell cycle is regulated and what happens if the regulation goes wrong. So for starters, let's talk about how it's regulated. The way that cells are told whether to continue the cell cycle or not has to do with chemical signals. Chemicals that they make that tell them to move forward. If they do not get a signal telling them to move to the next phase, then they will stay in the phase that they're currently in. And this is on purpose. These are checkpoints to make sure that everything is ready for that cell to move to the next section. So the key thing really is going to be this protein, cyclin. What cyclin does, when a cell makes cyclin, cyclin activates specific enzymes called CDKs, cyclin-dependent kinases. Now the enzymes, the cyclin-dependent kinases, are always there, but they're in an inactive form. So they're just sitting there, but they're not doing anything. When cyclin binds to the kinase, it turns the enzyme on, and the enzymes then, what a kinase does is it adds phosphate to things, and that's what this says here. So for example, at the end of G1, if a cell makes cyclin, the cyclin will tell the CDK to basically, it'll activate it, and the CDK will put phosphates on the proteins that basically start the DNA making a copy of itself. In other words, that starts synthesis. If there's no cyclin, then the cell will not move to synthesis. Instead, what that cell will do is it will actually go into G0. And I mentioned G0 yesterday. So G0 is sort of this stage off to the side for cells that are not going to divide. And you have some cells in your body, like brain cells, that are always in G0. They are basically shut down. They do their jobs, whatever the jobs are, neurons carrying information and all that, but they're never going to progress to S or G2 or make copies of themselves. So again, cyclins are the key to this. Cyclins are the proteins that activate the enzymes, and then those enzymes add phosphates to other proteins that move the cell to the next stage. Yes? Muscle cells, skeletal muscle cells are multinucleated. So their nucleus divides, but the cells don't. So you're actually born with all the muscle cells you're ever going to have. They can get bulkier and whatnot if you work out. But that's why some people would never get like really thick and bulky, because you're born with a certain number of muscle cells and you're not going to increase them. Yeah. Heart cells, for example, though, I think are in G0. So their nucleus isn't even dividing. Those are single nucleated cells and you have a certain number. Then you just put fat in between the muscle, or you put fat on top of it. But no matter how you work out, you're going to just build fibers between the muscles, but you're not actually going to make more muscle cells. You actually don't make more fat cells either. You're also born with all the fat cells you're ever going to have. It's just that if you eat a lot, your fat cells get bigger, and if you eat less, they get smaller. So some people are actually more prone to be heavier because they have more fat cells. So it's easier for those cells. That's why if somebody gets liposuction and then they just eat a lot, they'll just have like a big dent in that area where they had the liposuction. That area will never come back. That fat is gone forever. You don't make more fat cells. Yeah.
So to be in G0 is then the cells not dividing, not the nucleus?
No, technically the nucleus isn't either. When I say muscle cells here, I'm referring more to like heart muscle. Heart muscle, the nucleus isn't dividing either. Skeletal muscle, technically the nuclei are dividing. So I would say muscle cells, or skeletal muscle cells, are probably not in G0. Probably the best example would be brain cells of anything. I also know, and I have the next line here, and I know it's a little low and probably hard to see, that there are some cells, like liver cells, that apparently are in G0, but if they suffer a trauma, or you were to say donate a piece of your liver to somebody, that kind of thing, they can re-enter the cycle. So there are some cells that will go into G0, but they can come out of it. If we could, this is sort of some of the studies in how to solve the issue of traumatic brain injuries or spinal cord injuries. Can we play with these chemicals to make these cells reproduce and repair themselves? Because right now they don't. So you sever your spinal cord, it's done. Those cells are not going to reproduce and repair. Could we manipulate the cyclins and whatnot? Maybe. But it's also a little tricky because cancer is related to problems with this too, the other way, that the cells are reproducing too much. So it's not as simple as, oh, we'll just inject some cyclins and everything will heal. Any other questions about this slide? Okay, so that is in essence, this diagram will show you, this is just showing, you don't have to know this graph for a test or anything, but it's just showing you have different kinds of cyclins that stimulate the cell to go to the next phase. So you have one that's a G1 to S cyclin. There's one that starts mitosis. So those proteins stimulate specific CDKs. And this is showing here, this is the G1 to S cyclin. So the cyclin binds to CDK, activates it, and CDK then turns around and adds phosphates to these three targets. These are enzymes that actually start DNA replication. And adding a phosphate to things, this is why ATP carries energy. Because when the phosphate breaks off and gets added to something, it usually changes the shape of something, activates something, whatever. So anything that adds phosphate to things is going to cause those things to typically be activated or change in some way. So in essence, that's what's happening. Here's another diagram. This one's just showing again that there's typically a couple of checkpoints, a G1 checkpoint, a G2 checkpoint, and even a mitosis checkpoint to make sure that everything is okay. If the cell detects a problem, it should stop cell division, or it may even trigger the cell to self-destruct. It's called apoptosis, programmed cell death. Cells will self-destruct typically, or they'll send a signal out for the immune system to come destroy them. So that's what's supposed to happen if cells are not right. Cancer is exactly the problem with that. Cancer is where the cell is doing the opposite. It's not listening to the signals telling it not to divide. Some of the signals that would tell a cell not to divide, that would prevent cyclin from being released, one of them is called density-dependent inhibition. Basically, when cells are really crowded, they send signals to each other that stop cell division. That way those cells don't divide. You don't notice your skin getting all these lumps of extra skin cells. That's because when they're nice and full, they send signals to each other saying there's no need for division right now. As skin cells die off, they send signals for the cells to divide to replace those cells. The other one is called anchorage dependence. Typically, if a cell breaks off and floats around to some other location, it's not actually attached to tissue, it typically will not divide either. It will not receive the proper signals. Yes. Correct. Right. Oh yeah, almost all cancers, I think 99% of cancers are not something that you're born with, or not even genetic. Yeah, they're random. I'm going to actually tell you now what causes cancer. Question? Benign, it's a very gray area because benign can become malignant. That's actually a slide here too, but benign is what we usually call it if it's contained in one area and it's not actively spreading. That's what the term means. But if you read online, you'll see that technically benign things can turn malignant because you never know. So here's the reason why you get cancer. Cancer is nothing more, no matter what kind it is, brain cancer, bone cancer, leukemia, it's cells that are dividing out of control. There are two typical causes of cancer. One is that, for example, cyclin is being produced all the time because that's DNA controlled. So maybe a mutation that has happened that the cell just keeps making cyclin. It's only supposed to make cyclin when it's time for the cell to divide. But if a cell makes cyclin all the time, it's going to basically have that cell just dividing. And every cell it makes by mitosis is going to have the same mutation, so it's going to follow the same thing and continue dividing. Another thing it could be is the CDKs. You could have a mutation in the DNA of the cell that the CDK that's made, the cyclin-dependent kinase, is not made in an inactive form. It's supposed to be made in an inactive form that it's only activated by cyclin attaching to it. It's possible that the DNA mutates and this is made in a form that's already active. So now it doesn't need to receive a signal from cyclin. It just tells the cell to divide all the time. So the cell just keeps pushing through the cell cycle. What causes these mistakes in the DNA? Anything. Every time your DNA makes a copy of itself, it's possible that it could make a mistake. There's some things that increase it. X-rays, ultraviolet light, what do you call that? Asbestos, cigarettes, some of the things in cigarette smoke, certain foods you can eat, certain toxins you breathe. There's all kinds of things that can increase the chances of a mistake when the DNA copies itself. The other thing that could be the problem is this. One of the things your DNA codes for are literally what are called tumor suppressor genes. Their only job is to make enzymes that check the DNA and look for problems. If they find a DNA that's mismatched, it immediately sends a signal that that cell is damaged and that cell is supposed to die. But sometimes what causes cancer is not that you have a cell that has an overactive cyclin, it's that the tumor suppressor genes are missing. And that's actually how the HPV virus causes cancer. It doesn't directly cause a mutation in the cell that makes the cell cancerous. It basically destroys a protein that's involved in quality control. This protein, P53, is supposed to find cells with mutations and kill them. If your cells don't make P53, doesn't mean you're going to get cancer, but what it does mean is if you have a cell that becomes cancerous, there's nothing to catch it and kill it. So now the cell survives and reproduces. And that is how HPV and some other cancer-causing viruses actually go about causing cancer. They don't damage the DNA that makes the cell divide, they damage the quality control machinery.
So, shouldn't there be like a shot or something you get that could prevent cancer?
So there's not necessarily a shot to prevent cancer, but what we are seeing now are huge numbers of drugs that are coming out that specifically deal with these problems directly. Like old-fashioned treatment is chemotherapy, right? That kills every cell that's dividing. So it's going to kill cancer cells, it's also going to kill blood cells, and you're going to become anemic. Hair cells, your hair is going to fall out. All kinds of other cells are going to die, anything that's dividing. What the new drugs do is they actually look for the specific, we know specifically in some cases where the DNA is damaged, and these drugs find only cancer cells because we know we can make them look for a cell that, let's just say, has a damaged P53 gene, or we can look for cells that are missing or that have a damaged cyclin DNA, and it'll only kill the cancer cells. I would say in the next 10 years, it is very likely cancer will be cured because we're already curing certain cancers. If you look online, there are certain forms of cancer that we have found the cure. They treat it with a manufactured man-made virus, and that virus basically goes in and actively finds only cancer cells and kills them. It finds the ones that have the specific problems in their DNA that's leading to the tumors. So that's what's on the horizon, and I think that's probably, like I said, within the next 10 years, definitely. This is just a picture. Anchorage dependence, cells should be stuck to something to divide. Density dependence, when it's crowded, cells should receive signals not to divide. Cancer cells don't follow these rules. This is a picture, a micrograph of some cancer cells, and they're continuing to divide even though normal cells should have stopped as soon as it was a clear layer. And this is just the last little thing there. So cancer cells, typically your immune system also looks for abnormal cells that are cancerous. If it finds them, it will destroy them. But if it doesn't, that's where you get a tumor. A tumor is a mass of abnormal cells that's just continually dividing. Cancer cells don't make toxins. They're not eating other cells. All they're doing is dividing uncontrollably, which means they're basically choking out cells around them that are doing their normal jobs. And that's what a benign and malignant refer to. If it stays in one spot, it's called benign, a tumor. If it breaks off, which is called metastasis or metastasizing, that's a malignant tumor. But if you look online, it's actually a very gray distinction because it's not like if something's benign, it'll never break off and become malignant. If you have a tumor's a tumor. They're going to want to remove it because of the off chance. Because if a piece breaks off and it's not listening to density-dependent factors that tell it, or anchorage dependence, it could then settle somewhere else and then keep dividing. So that's what a tumor is, and that's what cancer is. I do think, like I said, the good news is that because we understand it a lot better now, we can find ways to cure it. So, and the HPV vaccine, obviously, what it does is it just prevents you from getting the virus in the first place. And the virus makes, certain forms of the virus, because there's a whole bunch of them, make certain things that then can deactivate those proteins that are cancer suppressor genes. So, all right, I'm pretty sure this may be the last slide. I'm not sure. I think there may be just a picture. I'll go back.